Afternoon, everyone. Welcome to another Statistical Society of Australia Biostatistics and Bioinformatics webinar. I'm Alicia De Oliveira. I'll be your chair today. So firstly, on behalf of the Statistical Society of Australia, in the spirit of reconciliation, I would like to begin by acknowledging the traditional custodian of country throughout Australia and their connection to land, sea, and community. We pay our respect to their elders, past and present, and extend that respect to all Aboriginal and Torres Strait Islander people today. Today, we are very grateful to have Professor Melanie Barlow speaking to us. Professor Barlow is a bioinformatician and a statistical geneticist with over 20 years experience. She's the team leader for Healthy Development and Aging at the Walt and Eliza Hall Institute of Medical Research. There, she oversees the scientific strategy for 3 divisions, including the Population Health and Immunity Division, which she co-established in 2015. Her research focus is the understanding of the genetic basis of human diseases, and her innovative analysis methods have led to major genetic discoveries. She has been recognized for her contributions to science as a winner of the Australian Academy of Science Moran Medal and the Genetic Society of Australia's Ross Crozier Medal. Today, her talk is titled Inferring Genetic Relatedness to Identify Disease Cause Invariants and Selection Signals. So during the presentation, you're welcome to post your questions in the chat. These questions will be presented to Melanie at the end of the presentation during Q&A. So with this, I'll hand it over to you now, Melanie. Thanks again so much for presenting.
Thank you, Alicia, and thank you for the invitation to speak. I don't get much time these days to attend Statistical Society of Australia meetings as much as I'd like to, so it's an absolute pleasure and honour to be invited to speak. Would you believe it? We have first hiccup already. Hopefully that'll work. All right, fixed that one. All right, that's WeHo, by the way, in the background. So when Alicia asked me to speak, she gave me a brief, but I did spend a bit of time thinking about what I should talk about. She asked me to speak about potentially papers on which I've published with summary analysis with code that are useful for biostatistics and bioinformatics in practice and perhaps general guidelines. So I thought a bit about what I should speak about, and then I thought, well, you know, I started my career and I did a PhD in applied probability in the statistics department at Monash, and really the overarching thing in my applied probability work, and which I still work on and come back to all the time, is this idea of relatedness that really has spanned my entire career. And I started my PhD in population genetics at Monash and finished that, in 1998. And I did it in a field called Coalescent Theory. And the Coalescent is a wonderful, quite complicated applied probability model that models long-term evolution of genetic material in populations. And right at the top here, you can see that this is a coalescent pedigree from one of my papers with my PhD supervisor. And then when I got to WeHo in 1999, when I joined Terry Speed's group, I started working in statistical genetics, so on pedigrees here. And pedigrees are really quite short-term timescale relationships between samples due to relatedness. And then in the last 20 or so years, I've worked a lot in linkage analysis, done some work in genome-wide association analyses, and really getting back into that quite a lot now, and have done a lot of work on next generation sequencing data and genomics. So all of these have got this overarching theme of relatedness, and it's the lens through which I view any samples, and that's just due to my statistical training and my original PhD work. So this talk, warning, is going to be a bit of a historical career reflection talk as well, but I hope to convince you that what I'm going to talk about is still highly relevant today, and hopefully maybe some of you will try some of these techniques that I'm about to mention that might seem old hat, but I don't think they are. So the overarching idea behind this talk is relatedness, and something called identity by descent. So let's define that in the kind of simplest way possible. Let's think about a haploid organism. So human beings are diploid. So this is a much simpler version. It's not really a human pedigree per se, kind of a subset of a human pedigree. What we're talking about here is, in particular, I want you to look at the bottom two individuals, and they share a common ancestor. And when you look at their genome, so this is effectively kind of one chromosome, as you walk along their chromosome, you can see that they share a fragment of their genome, and that's called, they share it identical by descent, because that green fragment is inherited from a common ancestor, the green ancestor haplotype at the top here. And these fragments are of great importance to us for all sorts of applications. So the changing of the colors here are due to the recombination process. So whereby our chromosomes are broken up during meiosis and mixed up. So, you know, all of what I'm talking about today is pertinent to organisms that recombine, which is not everything. And we'll return to that concept at the end of the talk. So, genetic data. Let's say we have some genetic data on a group of 6 individuals who are actually related in a pedigree over here. Mum and dad, so females are always circles, males are squares, and they have 4 offspring here. And here I've got some data from a SNP chip or something, and here I've listed the 6 individuals. So each row here is one individual from the pedigree. The first 2 rows are the 2 parents, and then the next 4 rows are the 4 offspring siblings. And here are the SNPs. So we have about 10 SNPs here, just a subset of the type of data set that we normally get. These are ordered in their positional location on a genome using information from the Human Genome Reference. So we know where they sit in relation to each other. These numbers in here are 0, 1, and 2, and they denote the number of non-reference alleles that each individual has. So 0 would be 0 non-reference alleles, 1 is 1, and 2 is 2. And you can also have missingness that would be annotated with a -1, but there's no missingness here in this story, just to make it simpler. And my first point is that genetic data, this matrix of data, has got a lot of structure in it, and that's due to various facets to do with the genetics and the origin of the data. So first of all, because these individuals are related, the rows are not independent of each other. You know, so here, if we, for example, look at the 3 rows here, or the fragment of data here from these 3 individuals, they're 3 siblings, three of the siblings, of the 4 siblings. And of course, they're correlated because they share the same 2 parents, but they've received different rearrangements of their parental genotypes. But sometimes, so going throughout the genome, they have they sometimes coincide to have exactly the same fragment inherited by descent. So there's lots of correlation between the rows. But then there's also correlation between the columns, and this is simply because of the way we inherit our genetic material from our parents, which is usually by chromosomes, and then recombination breaks up those chromosomes in fairly large blocks. And that's referred to as linkage between markers. So there's no IID here between individuals or SNPs. There's lots of correlations, but the good news is we can model that with statistical applied probability models. Okay, let's go one step further. So before I just showed you the pedigree, nothing to do with disease or anything, let's add in a layer of information. Let's say the 4 siblings are all affected by some disease. So that's annotated here with the individuals colored in black, and the parents are not affected. So we can add in a new vector on the far right-hand side here, which has binary. So these aren't SNPs in effect. They're the entries here annotate whether the individual's affected, annotated with a 1, or unaffected with a 0. Okay, so we are often interested in trying to find a relationship between a SNP in the genome and the effectiveness status of the individuals in the pedigree. And the reason we want to ask that question is because we know from the rules of genetics, we can use that information to try and localize where the disease-causing variant that causes the disease for this family might lie. And in a very naive way, you can just do that by basically looking for correlation between the SNPs in your genotyping data from the individual and the column on the far right-hand side, which annotates the disease status. So we could go through marker by marker and simply ask, what's the relationship of this data with our data on the far right-hand side? And can we just develop a fairly simple score and measure that relationship? And that is in effect what we do in linkage analysis. We develop some kind of score, we move through, and then you can see, because I've manufactured this example, this block in red has got 4 markers in a row that are in linkage that have strong correlation between the effectiveness status and the genotypes. And that would be then reflected in our test statistic. So, you know, that would be a very naive way of doing it. So just before we get into the kind of mechanics of the statistical model, I just want to say something about the types of marker data that we use these days. So in order to perform these types of linkage analyses, you need roughly 15,000 SNPs that are roughly uniformly distributed throughout the genome. You preferably also want SNPs that have got a high minor allele frequency because they are more informative for linkage analysis. So where could these markers come from? And this is something that people don't realize, but these days we would be using SNP chips. So the latest and greatest SNP chip that's being used worldwide now is the global screening array from Illumina. That's got about 600,000 SNPs. So we definitely don't need 600,000 SNPs. In fact, that would contravene some of the aspects of the model, of the statistical model. So we pull out about 15,000 good SNPs, and I'll refer to some software to do that. And what's really interesting is how cheap these chips are now. So they are roughly, I mean, if you purchase them in bulk, about $30 Australian, which is fantastic. So you can really do these studies very easily now. But you can also pull out what we call pseudo SNP panels from any kind of next generation sequencing data, whole genome sequencing, whole exome sequencing. You can even do it with RNA-seq data because you can still call SNPs within that. And we have a number of publications that really explain how to do that. The Human Genetics reference there is an overview of all of these techniques. And the Genome Biology paper from 2011 explains how these pseudo SNP chip panels can be formed and the software that's needed. So let's go back to the actual algorithm that's needed in order to perform linkage analysis. And we really want to utilize the wonderful statistical frameworks that serve us so well in many of our other statistical endeavors. And the test statistic of choice in linkage analysis is something called the LOD score, which is really just a transformed likelihood ratio test statistic. And there we compare the null hypothesis of our genotype matrix from our pedigree, G, and the disease vector, D, being unlinked. So unlinked in this context means that they're segregating independently. And that's the null hypothesis. We compare that to the alternative hypothesis of G and D being linked. And the way you do that is you calculate the likelihood ratio test statistic at various positions throughout the genome where you insert the vector, the disease locus vector, in amongst the columns of our genotyping matrix G and calculate our likelihood ratio test statistic at various positions. And that gives you a very powerful, classical statistical test statistic to score the relationship between your effectiveness status in amongst all your genotyping data to try and map the location of the disease locus. So the likelihood is really a probability calculation of the genotyping matrix G and the vector D, which describes the effectiveness status, conditional on your knowledge of what the pedigree structure looks like. So you are told exactly how the individuals relate to each other according to a pedigree that they're given. And that can be calculated with really elegant, exact methods that have been around for a very long time and have really, you know, have come to a halt in terms of development because they are, you know, really wonderful and they're done. So the most famous of these and the one most relevant to SNP data is the Lander-Green algorithm. And the best piece of software to use that is something called Merlin. And that was developed by Gonzalo Abecasas, who's a very good statistical geneticist. And we still use this program to this day. If the pedigree that's very large, then you have to jump to a Bayesian Monte Carlo Markov chain technology setup. And the huge body of Elizabeth Thompson, who's also a very famous statistical geneticist, now retired, her life's work is embedded in this package called Morgan, which you can Google both of these and download these very easily. Okay, so in a nutshell, or rather going a little bit deeper, the relationship between the SNPs that are in the genotype data matrix G and the disease locus D is modeled with a time inhomogeneous Markov model. But here, time is not actually the measure that you use. What we actually use here to describe the recombination process is the genetic map distance. And that is something that has to be empirically estimated, but is now available for humans and many other species. But you can also roughly approximate it with physical map distance if you have a genome reference for your critter of interest. So there's no need to, you can do this in any species that recombines effectively. So basically, the likelihood is set up and the calculations for the likelihood, even though we've got a very big matrix, a very complex set of structures, it's really utilizing the rules of genetics to formulate a hidden state. In this case, these are really kind of indicators of the status of recombination events that have happened in the pedigree, which I don't have time to discuss in detail. Utilizing the rules of genetics and this hidden state leads you to be able to efficiently factorize the likelihood. And all of this is embedded in a framework that's now very well known as a hidden Markov model. Hidden refers to the hiddenness of the actual underlying kind of recombination events, and hence the inheritance states at different loci in the probability model. And it's a Markov model because we're modeling the relationship between SNPs with this time inhomogeneous Markov model. But this type of thinking, which I've just described to you in the linkage setup, which is, you know, a beautiful, elegant model, well known, very old now, this type of thinking carries across to all sorts of other probability models in genetics and across to other relatedness applications. So let's jump to a model now where we don't have a pedigree. So this happens all the time in GWAS. GWAS cases and controls are ascertained usually under the assumption that they're independent, that they're unrelated. But they could indeed actually be related. And for GWAS analyses, especially for the pre-2015 GWAS methods, relatedness was actually a nuisance. And what you can do with GWAS cohorts is you can do an analysis whereby in a pairwise manner, you take all your pairwise combinations of all your cases and control individuals and ask whether they may be related. And if they were, people would prune those individuals, well, one member of the pair out because they went against the assumptions of the GWAS test statistics that were used pre-2015. So either, for example, Fisher's exact test, where you've got independent, identical distributional assumptions, or, for example, logistic regression models, again, where in that framework you were assuming independence between samples. As of 2015, GWAS methods have really improved with now you are able to incorporate related individuals as well through methods such as LMM Bolt and SAGE. I'm sorry, I didn't stick the references in there because that's not the main focus of the talk. But prior to that, people used to prune related individuals according to some empirical threshold. What are some other applications where you might want to detect relatedness between individuals? Well, there's lots of old agricultural or zoo applications. For example, in Melbourne Zoo, for example, you might have cohorts of animals that are kept in a small enclosure where you might have multiple males, multiple females, and you don't know who's made it with whom. And it's important to figure out what the mating patterns were in order to make decisions to ensure that your local zoo population is as diverse as possible to, of course, prevent nasty inbred diseases, etc., etc. So diversifying your genetic pool is really important. So that's also an application. And as you will see in the talk, detecting relatedness, even when you don't have a pedigree, is incredibly powerful for human disease studies. So we'll get to that. But on the whole, these methods where you don't have a pedigree are referred to as identity by descent methods. Okay, so just going back to these identity by descent methods, recall your pedigree from before. Now it's like we can see the pair of individuals, but we don't know anything about the pedigree. But we are going to develop statistical models where we try and identify relatedness between these two individuals, which is driven by regions in the genome where they still share a fragment inherited from an ancestor identical by descent. But we don't know the pedigree, we're just inferring it's there. Now, just another general comment, we don't know the exact nature of the pedigree, but the depth of the pedigree, the number of meioses that separate these two individuals is important for our models. And that also, because that determines kind of the rate of recombination, but it also determines the number of these identical by descent fragments that are left. Obviously, the more distant the common ancestor is, the fewer fragments are left, and the smaller the size of the fragment. So these are really important concepts. Now I need to introduce you, when you go to this inferring relatedness between pairs of individuals when you don't have a pedigree, you also need a concept of global versus local relatedness. And that harks back to what I was saying before. Without a pedigree, we need to get an estimator first up about some kind of global or genome-wide relatedness. Roughly how far apart are these two individuals? And the reason we need to know that a priori is because when we develop our model where we actually want to identify the actual fragments that are identical by descent, the time inhomogeneous transition probability model is dependent on the number of meioses between the two individuals. So the way that's done at the moment is that's calculated with a method of moments estimator based on just based on using counts of alleles that are shared identical by state. So just kind of scoring how many alleles are identical by state. And from that, you can develop method of moment estimators for global or expected values of identity by descent. And I've just mentioned two pieces of software, King and Plink, which you'll see the references of later on, that will do this calculation for you. And you'll see an application soon. So just before we go into the first application, I just wanted to put this kind of work into the context of my own academic journey. So when I first came to WeHi, I worked on my first IBD model, which was never published, in 1990, where I worked on an IBD model in siblings without parents. And back in those days, we didn't have SNP chip data, we just worked on microsatellite data. And I spent about a year developing the model, and I didn't publish it. And I think that was a really salient lesson for me, that from my field where I previously worked in my PhD in Coalescent Theory, things moved fairly slowly. There's, a fairly boutique group. When I moved to medical research and statistical genetics, it was a much more active field. And I learned a very harsh lesson that a lot of work that I did, I ended up getting scooped in that model by somebody who developed effectively an identical model and had written it up earlier than I did. So that they published and I didn't. So that was kind of a big lesson for me in the field. So there was some work on IBD models and applications thereof in the middle, but really the biggest next application or insight from my group was this 2011 publication, where we really described software that really showed that you can pull out these pseudo SNP chips and then apply all of these wonderful tools for relatedness and so on with the data that you can pull out of what was then starting to become a really huge burgeoning field of next generation sequencing applied to, in particular, Mendelian disorders. And then, you know, this is all backed by work by wonderful PhD students. So that work was from a PhD student, Catherine Smith. And then Lyndall Hendon, who was another fabulous PhD student, did some nice work in 2016, where we developed an identity by descent model specifically for the X chromosome, which hadn't been done. And we applied that in a variety of applications. So that's kind of, so I'll stop at 2016. We'll come back to that journey later. I now wanted to go into the first application just to show you the power of what seems like really old-fashioned approaches. So this paper was accepted yesterday by chance. This is work by Karen Oliver, who started a PhD this year in my group, but also did a Master's of Bioinformatics with me many years ago. So that red arrow is pointing to Karen at our 2019 Barlow Lab Christmas lunch, and I think that was our last get together before we all started working virtually due to COVID-19. So I like looking at that picture. Okay, so Karen worked on this these two pedigrees. So these two pedigrees, one is from Portugal and one is from Turkey. They have 6 affected individuals in them who segregate a very nasty epilepsy called progressive myoclonus epilepsy. And these were studied, as you would these days, with exome sequencing. Most PMEs are caused by recessive mutations, and a very promising recessive mutation was found, but it was in a novel epilepsy gene that had never been described before for epilepsy. And the variant looked very promising. It was ultra rare. It was a homozygous splice altering variant, and subsequent RNA analysis with RT-PCR confirmed that it was a splice altering variant. So it looked promising, but the gene, as I said, nothing known about it. And these pedigrees and, you know, the methods that we got, we could potentially use them to gather more evidence to support this variant and this gene as being a truly putative, truly novel epilepsy gene. The issue with these rare disease studies is always that this is as good as it's going to get. You have 2 families worldwide who are segregating this ultra rare variant, and you might never see another family. So it's very hard to get enough evidence together to convince people that this is a truly novel gene for epilepsy. I should also say epilepsy is kind of the poster child for rare Mendelian contributions. There are over 100 known epilepsy genes by now, and more being found all the time. Epilepsy is a very broad umbrella. So just something else about these pedigrees, you've probably noticed that there are double lines here. When you see double lines like this, that indicates that the parents are inbred or they're consanguineous. So that means that the parents are related. And you can see the Turkish pedigree here. There are all 3 couples here are reported to be related. The Portuguese family here, there's a dashed line indicating here that the parents thought they may be related, but they weren't sure. Okay, and that's really important in the context here that the putative disease-causing variant annotated by little v here is homozygous in this pedigree, and it's exactly the same variant in this pedigree. So the only way that could have happened is if in both pedigrees, the variant has flown down through both sets of parents homozygously. And similarly, if they share the same variant here across the 2 families, either these 2 families are related and have a common ancestor, or it's a recurrent mutation. So these are all big questions. So we would like to gather more evidence for this variant and gene as a novel cause of PNE. The rare mass suggests this variant is inherited homozygous recessively by both parents. Can we find further proof of this? If we can find statistical proof of this, and this is a really low chance event. And if this information coincides with our variant, that would really pump up the significance and the likelihood that this variant is indeed the causal one, because we're putting together two kind of separate lines of evidence. So I've already mentioned this. We really needed to know that family 1 was truly consanguineous, not just self-reported. Are family 1 and 2 related? And could we potentially find these regions here? These are not regions that are identical by descent, but they're more than that. They're homozygous by descent within both families. So it's a slight tweak on this identity by descent context. Okay, so we had SNP chip data for 9 individuals from family 1 and 6 from family 2, and that's annotated by the little hash next to the individuals in the pedigree. So quite a nice set of data. And this is kind of our standard kind of computational pipeline or things that we throw at pedigree studies such as this. We do our QC and data prep with our software, Linked Data Gen. We do an inbreeding analysis with something called FSTIM, and that was developed by Anne-Louise Leutenegger, who did a PhD with Elizabeth Thompson, whom I mentioned previously. And this is another hidden Markov model utilizing very much the same framework as I introduced you right at the start of the talk. But this time here, you're looking for regions that are homozygous by descent. We performed these global relatedness checks with these 2 pieces of software that I've already mentioned, Plink and King. And we're going to do that within and between families or individuals that are from within and between families. We're going to also do a proper linkage analysis, again, with our hidden Markov model approach embedded in Merlin. And then we're also going to visualize the haplotypes, although I won't show you the results from that. So the first thing is this analysis with this FSTIM piece of software. So that estimates inbreeding coefficients in these pedigrees. So given particular relationships, you can estimate the expected value of F, the inbreeding coefficient. So F is the probability of being at a particular point in the genome, and that being homozygous by descent. So if the parents are first cousins, then their offspring, as you go along the genome, has a 6% chance of that point in the genome that you're looking at being homozygous by descent. As the parents' relationship gets more distant, that probability decreases. And then by the time you get to third cousin, there's not much probability left. Okay, so this nice piece of software does this for you, and sorry, it's a little bit messy, but the bottom line is Karen was able to recapitulate that indeed the Portuguese family, all the purported inbreeding relationships were true. And the one that was reported not to be inbred here, also we didn't find any evidence of inbreeding. But she could also estimate roughly what the relationship between the parents was. And you can see that they're fairly distant. Whilst in the Turkish family, it was kind of quite more confronting. The relationships, which were also reported, were much closer between the parents. And this relationship in particular is quite close. And so that leads to much higher inbreeding coefficient estimates here in the Turkish family than it does over here in the Portuguese family, but a lot of inbreeding overall. So really supporting that this variant was inherited homozygous throughout both families. But we still haven't said anything about the relationship between the 2 families. So now we'll turn to Plink, which allows you to estimate global relatedness between pairs of individuals. So it estimates this probability vector for identity by descent, so we won't worry about that too much, but it estimates also kind of average IBD, or the proportion of IBD, this pi hat vector at the end. So let's just pay attention to that. We won't worry about these. So if 2 individuals are unrelated, then if 2 individuals are unrelated, then they have a pi hat of 0. And if they're identical, so here X are the same individuals, then they have a pi hat of 1. So that would be applicable to monozygous twins, duplicate samples, etc. And down here, not related. So, and then you have a gradation of all these relationships in between for these results. So you can estimate pi hat from Plink and have a look what that matches up to in terms of estimated versions of pi hat given a pedigree. Okay, so Karen ran this analysis for our 2 different families. Remember, family 2 was the more inbred Turkish family. And here, these are just the results from the pi hat test statistic. And you can see here that basically she got pretty much what she expected. So the dashed lines here are, you know, 1st degree, relative 2nd degree, 3rd degree. So, the only thing to say about the results from the Turkish family is that in general, all of these were inflated, and that's due to the inbreeding in the pedigree. We see that quite often when we have heavily inbred pedigrees. The pi hat estimates are higher than what you would expect because we calculate the pedigree relationships under no inbreeding. She also did this for family 1, which is the Portuguese family, and they are sitting much closer to the expected values, less inbreeding. But all of these relationships fit. But then disappointingly, when she looked at individuals where one individual's from pair 1 and one individual's from pair 2, she didn't see any close relationships. And really remembering that one family's from Portugal and one's from Turkey, you know, that's not unexpected. Now, I've got to point out that that does not mean that the variant is not potentially inherited from a common ancestor. It just means that that relationship is not recent. So up to, you know, a distance of 6 meioses or 3 generations, or at the most 4 generations up to a common ancestor. It's just that this test statistic, it does not have a lot of sensitivity once you get beyond that sharing. Okay, so from our own experience with many of these tools, the best strategy to then go forward is to recode your pedigrees with what you've learned from your detection exercise with the inbreeding analysis and the relatedness analysis, and recode the pedigrees to incorporate the new information relatedness. So these pedigrees are kind of fixed up versions. And you can then, you then have to import all of your genetic data, your disease locus, information on effectiveness status, and the pedigree structure into this program called Merlin, which will perform the likelihood ratio testing, so the linkage analysis. And here are the results for the linkage analysis. If you remember, the LOD score is our likelihood ratio test statistic. LOD score of 3 is about a chi-squared likelihood ratio test statistic of about 12 and corresponds to genome-wide significance, so a very low p-value because you have to adjust for multiple testing here. So 3 is generally regarded as the threshold for genome-wide significance. So you can see from this analysis that we have got really fantastic results for both families. Both families have identified a statistically genome-wide significant peak on chromosome 16. So family 1 is in blue, has a much narrower peak there, and family 2 is in brown. And best of all is that they overlap each other. And just zooming in on that a bit further, you can see here family 2, which is the Turkish family, gives a much broader region, a sort of 30 centimorgan region, so roughly 30 megabase region of linkage, where it says the disease-causing variant must be contained within that. And family 1, which is more informative and more outbred, the Portuguese family, narrows that peak even further, but it is contained within the other one. So thinking about this and what this region represents, it is this region here, meaning basically the intersect between these two is just a region from family 1. That's about 10 centimorgans in length. The human genome is about 3,600 centimorgans. So you've narrowed the region where the disease-causing variant lies to 1/360th of the genome. And if you saw here, Karen then, of course, had a look where our putative variant sits, and it sits right in the middle of that peak. That is fantastic. So the chances of finding this coinciding is highly unlikely. And then the last thing that I won't show you in detail is then we also went back and asked, even though we couldn't find any close relatedness, we had a look at the haplotype on which the variant sits. So that's more of a sharing just by state. There's no IBD model involved there because of the scale and the time difference. We also saw that these two families shared the same haplotype, and it's slightly less than 1 megabase. So, whilst we have no proof, it is conceivable from what we know about human genetic history that an ancestor from Turkey may have ended up in Portugal at some stage, but probably a long, long time ago. And this rare variant has stayed around in these two locales in the region in Turkey and in the region in Portugal, and because of their own separate founder effects, have managed to stay around in those little subpopulations, albeit being very rare overall in the human population, and have caused these incidences of recessive disease. Okay. So that was our human application. So I hope that's convinced you that, as I said, that's just been published. So the other application is a non-human application. This is now already 2 years old, but it's a great application, I think, because you can do this to other species as well. So this is work by Lyndall Hendon, who's now at Macquarie University. Lyndall was a Choose Maths ambassador. So in lieu of finding a nice picture of Lyndall, I thought I'd put her big picture on the bus up here, which is very nice. So, right. So we are going to be talking about the pathogen that causes malaria. So I know we're all preoccupied with COVID-19, but malaria hasn't gone away. And not this year, but, previously it kills many hundreds of thousands of individuals every year in order of kind of 500,000, with many more cases of malaria. And it's been around for a very, very long time. Most of the deaths occur in children under 5 years of age. And malaria is caused in humans predominantly by two species, Plasmodium falciparum and Plasmodium vivax. But the story today and the data I'll show you is just from Plasmodium falciparum. So for the purposes of our talk, it's important to recognize that Plasmodium is a protozoa. It's a unicellular eukaryote. It's not a bacteria. It's not a virus. It's a kind of a fairly simple organism. So the falciparum genome, when we look at it, it's got a predominantly haploid life cycle. It's got about 5,300 genes. The genome is much, much smaller than the human genome, like 1/100th of the size, 14 chromosomes. It's got a nasty low GC content, so very high AT rich genome, but it still works for what we're about to do. You can still do many things with it. The important thing for our relatedness story, etc., is that it has a diploid phase and it recombines. So we have been working in malaria for quite some time, and you can ascertain the pathogens by taking blood samples from infected individuals. So you're taking the samples from individuals, but individuals could be affected by more than one malaria infection, meaning genetically different organisms as well. So we would like to do some work about asking whether pathogens in two individuals, whether they're related. We're not asking whether the individuals are related, we're asking whether they're pathogens, the Plasmodium falciparum that's infected these two individuals, whether they're related. And the complication here is, of course, as I just mentioned, some individuals have more than one infection, and so this becomes a lot more complicated. So Lyndall wrote a beautiful hidden Markov model, which we call isorelate. It's not just for falciparum, it can be used for any species where this kind of phenomena applies. So it works for P. vivax as well, the other human disease causing malaria pathogen. There you go. There's the Markov model, and there's the hidden states. And again, we want to find the regions that are identical by descent. She applied her model, which took some time to develop, to a very large data set from the Malaria Gen Consortium, where we had nearly 2,400 isolates, so samples of the plasmodium pathogen, and 42% of these data sets have got what are called multiple infections, so where there's more than one pathogen, and we can't pull them apart. We're dealing with a pool, so that's really interesting. So not only can we do pairwise analysis of pairs of draws of the pathogen from different individuals, we can accumulate those identity by descent relationships and sum them over cohorts. And when you do that, what happens is you accumulate regions of the genome where there's peaks of very close relatedness. What does this have to do with pathogens? Well, it so happens that there's huge problems in the pathogens that cause malaria in terms of drug resistance. So chloroquine, the CRT locus here, is a well-known drug resistance locus, as are these other ones, but we'll focus on chloroquine, where there's been a lot of evolution by the organism which combats a previously very effective anti-malarial called chloroquine. What we can see in different countries here is that some countries have very strong signals of selective pressure. So where the pathogen has reacted to this ongoing exposure to the drug to become clonal for resistance markers, for resistance alleles, leading to a decrease in efficacy of the drugs. And this is really important to know, because the drugs are our frontline defense for malaria. There's still no decent malaria vaccine. So knowing where these drug resistance loci sit, so you can map with these, and knowing the extent of the drug resistance and what individuals carry them, which you can all do through this identity by descent process, is really important. The other neat thing you can do is also ask about global relatedness. And here you can see that there's some very close relatedness between particular isolates. And then lastly, you can look at local relatedness. And here you can see that we have one founder event in particular that's represented in this data at chloroquine. That's marked also, if you look at the genome sequencing data, by this mutation. This mutation is not recurrent. It arose once and has spread everywhere around the globe to these different countries. Just one point to make, Papua New Guinea has an independent generation of this mutation. That is a recurrent mutation, but it's got a different origin to these main ones. So that's a lovely application of IBD in pathogens. So just to complete my academic journey, I just talked about isolate. Things are getting cheaper all the time here. We're currently working on a new IBD model with another PhD student, Randy, so I hope to tell you about that in the future. I love my relatedness work. It's spanned my academic journey, but it's not always an appropriate method. And I just wanted to point out that TB and SARS-CoV-2, the thing that causes COVID-19, are not appropriate in this setup. They don't recombine. These models are pointless for these organisms. The other negative about any of these IBD methods are that they're very time consuming to set up. And I'm really running out of time. I've just got a bit more. I just wanted to point out, probably many of you are really familiar with companies such as 23andMe and Ancestry.com. And they're probably more famous for their ancestry tests. So you've probably, maybe family members or you yourself have done those tests. And they're very fun because you can determine how much Neanderthal is still represented in your genome, how much Italian is in you, whatever you like. And they're fun to do. But these companies also offer to find genetic relatives for you. And the way they do that is through these method of moments estimators. So they basically look at global or genome-wide relatedness. And again, as I have showed before, really they lose their sensitivity due to the dropout. You run out of, the probability of finding a region by chance is very low once you get to relationships that are more distant than third cousins. But these techniques really work, and they have permitted many individuals around the world to find genetic relatives. So there's also public versions of this, and one of these is called Jig Match. And that's become really famous because you've probably heard about the story of this terrible mass murderer in the USA who was known as the Golden Gate Killer. And in frustration with not being able to solve this case, but despite having ample DNA evidence from the crime scenes, but the killer not having their profile in FBI databases or whatever, they in frustration turned and put the profile on Jig Match and used that to triangulate, because that's what effect you're doing, you're genetically triangulating relatives of your killer who are in these public databases, and from that you can go back and infer the identity of the killer by putting that together with your knowledge about where the crimes were perpetrated, etc., etc. And that's now been done not just for the Golden Gate Killer, but for I think more than 30 killers. I think 30 serious crimes that I read that number in an article recently. Sorry, I couldn't find the article again. But it is an ethical privacy minefield, and it's being heavily debated as to what this means for relatives who have their data on these public databases. It's really interesting. Look, if you're interested in this IBD stuff and the 23andMe, then I would recommend this really nice, relatively recent book by Libby Copeland from the US, and she's written a book called The Lost Family, and the subtitle is How DNA Testing is Upending Who We Are. That describes a lot of the stuff that I've talked about here, but in a kind of more soft science way, and describes it very nicely, but also really goes into the ethical minefield that this kind of work leads to. It's a really interesting read. I was quite enthralled, even though I understood a lot of the science already. So that kind of brings me to wrapping this up. So going back to my brief, what Alicia gave me, I hope I've done a few things that I was told to do. So I just want to point out our software is on those two links, the bits and pieces that I mentioned today. And I just wanted to, again, highlight work of people, past and present members in the Bala Lab who've worked on various aspects of identity by descent models. So, yeah, thank you.